Today's lesson is on the cytoskeleton. The cytoskeleton has a couple of different functions, and there are 3 different types of cytoskeleton. The function is to maintain structural support of the actual cell, the shape of the cell. It provides anchorage for the organelles. It is responsible for cell locomotion, so the cilia and flagella structures that some cells have to move around, those are made of cytoskeleton. And for regulation, they organize structures and activities of the cell. They're actually responsible for providing kind of a network for all of the organelles and anything that needs to move throughout the cell. So the cytoskeleton has a lot of different functions. Structurally, there are 3 main protein fibers that it is comprised of: microtubules, microfilaments, and intermediate filaments. And each of those has a different size, and the size works with what its job is. So here's a couple different images here. These blue tubes are the microtubules. These purple things are the microfilaments, and then the intermediate filaments are the green kind of mesh that's embedded throughout the cell. From an evolutionary perspective, proteins that make up the fibers are very similar in all living things, all the way from bacteria to humans. So that indicates that these fibers, these protein fibers of the cytoskeleton, are very ancient. They have evolved very, very early on with the earliest cells, and that they are essential for life. So the microtubules are the first one that we're going to talk about. They are the thickest of the fibers. They are hollow rods constructed of a protein called tubulin, and they can grow or shrink as more tubulin molecules are added or removed. And you can see their structure here. You can see the chain of amino acids, and then it's all coiled up into this little tube. The job of a microtubule is to maintain structural support and for movement inside the cell. They move the chromosomes around during cell division. This is what the centrioles are actually made of, are microtubules. They provide the tracks that guide motor proteins that carry organelles to their destination. So if you have something that's being exported out of the cell, it would be carried along a microtubule in order to make it to the cell membrane so it can leave the cell. And then cilia and flagella are both made of microtubules. So the centrioles are responsible for moving the chromosomes during mitosis, cell division. When you learned about them before, we talked about how they looked like little pieces of pasta. They extend and contract so they can grow and shrink back down as necessary to move those chromosomes. Cilia and flagella are extensions of the cytoskeleton that go outside of the cell membrane. Cilia are numerous. They are a bunch of small little hair-like projections that would surround an entire cell. And flagella are 1 to 2 extensions, and they're longer, kind of like the flagellum of sperm cells. Cilia provide an oar-like movement in order to move a cell in whatever direction it needs to go in. And flagella have kind of a whip-like motion, and they would propel the cell forward. Microfilaments are the thinnest of them. Okay? They are tiny, right? Micro means small. So microfilaments are very, very small structures, and they are a solid rod of a protein called actin. That it's 2 of them that are twisted together. They provide this 3D network inside of the cell membrane, and they interact with muscles to create muscle contraction. So the microfilaments are located all around the inside of the the cell membrane, and they actually help with contracting of the cell, moving of the cell. Some single-celled organisms like amoebas here actually move based on these contractions. This is also what splits an animal cell during cell division, during cytokinesis. These fibers that run just underneath the cell membrane are able to kind of pull and contract the entire membrane of a cell. And then the intermediate filaments are the last of the cytoskeleton structures. They are the intermediate size, they're in between. Their job is to hold things in place inside the cell. So they create this kind of crisscrossed network throughout the entire cell. Okay, the orange is the nucleus, and all the rest of this is the green is all intermediate filaments. So it's a big mesh that holds all of those organelles in place so that they're not just free floating around inside the cell. And again, here's the diagram. You've got the microfilaments just underneath the cell membrane. You've got the microtubules, are the big blue ones that transport things throughout the cell. And then the intermediate filaments are the green crisscross structure that holds everything in place. So here's a summary of all of that right there. The next part of this section is about cell junctions, where two cells come together. And you can see here that this is one cell on the top and another cell on the bottom. And there's this network here that's allowing those two cells to connect together. So plant cells have a mechanism for being held together. The structure of the cell wall is generally composed of cellulose. They do have a primary and a secondary cell wall with this kind of layer in the middle called the middle lamella. But cells in plants need to exchange materials, right? They need to get water from one to another. They need to get nutrients to go all the way from the roots all the way up, and those have to transport through cells. And if you look here, you can see there's little gaps in that cell wall structure. Those are called plasmodesmata. They're little channels in between the cell walls of two cells that allow the exchange of materials. Animal cells have this extracellular matrix that is kind of a variety of different, there's some sugar molecules, some carbohydrates, some protein, fibers. All of this network is outside of the cells. We have the cell membrane here. We have these proteins that are embedded in the membrane, and then underneath here you can see those microfilaments that are going to help move and contract that particular cell membrane there. But the junctions between animal cells, there are three different kinds. We have tight junctions here that kind of lock two cells together. It's almost like sewing where they get tied together. And they actually create little gaps there that allow materials to be able to pass through that cell membrane. And then you have these desmosomes here. Desmosomes are anchoring junctions. They fasten cells together in strong sheets. So these filaments extend out into the other cell and really provide a strong anchor between those two. And then gap junctions are another channel here that allow for the exchange of materials between them. So we have this tight barrier here. We have another anchoring barrier here, and then we have a channel between those cells. So here's the description of all of them for you. And my final thoughts today are some stupid penguin tricks. And that's what I'm going to leave you with today.